now we can go. So good morning, everyone. Thank you for coming. I'm glad to introduce you Dr. Mina Desai. Mina is from Harbor Medical Center from UCLA. And we have a project together. We are collaborating in this project since 2019, right? With also Kelly Coca from Unifesp. So we are so grateful and so happy to have you here, Mina. And I don't know if you know, if all of you know Mina, but she made her PhD in Cambridge with Dr. Nick Hales, who knows Dohut concept, know the works from Hales and Barker. So Hales were her advisor during the PhD, and Mina was responsible to spread this concept in US when she went to UCLA, and also here in Brazil. We have a history with Laís and Mina Desai. When Laís was doing her PhD with me, Laís said, I want to know Mina Desai. I want to work with her because she inspired me. And Laís was there for almost 6 months, right? Working with Mina in her lab. And when Laís come back to Brazil, we started this collaboration. So Laís is an important person in our lab to get Mina and Dr. Michael Ross to our group, to our team. So I'm so grateful to have you here, Mina. Thank you so much. Let me tell you, Mina said that she wants this lecture be informal. So you can you can ask if you have any question anytime. You can ask her. You can stop the class, the lecture. So to make you comfortable to do that. Okay. Thank you.
Can you hear me? Yeah. Okay. Thank you, Adriana, for that introduction. It is a great honor and a pleasure to be here. And like Adriana says, we have a great collaboration with Adriana, Márcio, and Kelly. And obviously, Adriana stole my thunder by telling the story, but I'm so sorry. Also, I would like to say, even I'm nervous giving a talk because I'm talking hoping that you understand what I say, okay? So if you don't, please let me know, okay? Okay, so this story started in 2017. As usual, it was a day when you go through lots of emails early in the morning, and one email caught my attention. And this has to also do with the talk that I'm giving today. This email came from a student that used the right words to catch my attention, okay? So.
She was sexing her day already.
Yes. So, okay, no, sorry. So this is what she said in her email, that she has read many of my papers. Of course, you know, everyone feels good if someone reads your paper. And then she said she heard my lecture, which said, oh, wow, this person did. And she used the word, I'm a big admirer of your work. Definitely, that caught my attention. And I quickly responded. And as Adriana said, it's Laís that wrote me this email. And I responded quickly and said, What do you need? How can we make this work? So when she was in my lab, obviously, I found her to be very dedicated, very pleasant, and when we were looking in Brazil for our research, her mentor came to my mind. I said, if a student is that good, a mentor has to be excellent. So that's my story. That's how I came to be here. Okay, let's talk about scientific writing. How many of you have already written a paper? Have you written, any of you written a manuscript? Yeah. How many? Just raise your hands. Okay, so you have quite a few here, right? Okay. Have you felt that it's a very hard work? And why do you have to do it? Right? You are a scientist. But as scientists, we first of all think, right? We design an experiment, we get an idea, do the experiments, we do the actual science, look at our data, analyze everything, and importantly, we have to communicate, okay? We do all these roles as a scientist. And if you don't communicate your work, it's not done, right? So it's always important to report and let the world know what you are doing. Otherwise, no one knows what you're doing. So that's why we write manuscripts, be it grants, be it review papers. Do you hear an echo, or is it okay? Yeah. Okay. So do you think of this? This is what I felt when I was writing my first paper as a PhD student. I would sit thinking, what do I write? What should my next sentence be? And then when I wrote it, I would like, oh my God, this is terrible. Okay? And then I would think, why can't I think properly? And sometimes I would think, oh no, I don't think I can ever do this. But everything comes with practice, right? Just as I said, that if you don't publish your work, no one knows about it. And I don't know about here, but I'm sure it's the same as in America, that our promotions, everything, it depends on how many papers you have published, how much funding you have got, right? So if people sit on my promotion committee, don't know me, but they can see a list of publications, right? So it's very important that you publish whatever work you do. So like I said, if you haven't written it, you haven't done the work. So key thing is publish. So there are different types of scientific writing. You know, you can write journal article, like, you know, on a topic, like an editor's notes. Scientific papers, you can write grant proposals, and oral presentations at a conference, posters. But I'm going to talk about mostly scientific papers today. And please feel free to stop me and ask questions anytime, okay? So when you start writing a paper, these are the questions you need to ask yourself. What is your paper about? Is my paper going to be interesting? And what will a reader or I gain after reading? Okay, let me. So as when you write, you have to think of your audience, right? So if I'm writing a paper, I would say in my community, like if Adriana reads, or Márcio or Callie reads my paper, if it's how can they easily understand in the paper, right? So you ask the question from the audience perspective, and then as a reader, would you read your own paper? You need to ask yourself that, okay? And from this paper, where do I go? Okay, so, and obviously, you learn more and more from what you write, right? Yesterday we had a discussion and we realized that at the end of writing that manuscript, you had more questions, right? So this is how science works. So before I get into really how to structure a manuscript, these are some of the questions and considerations that you take into account when writing a paper. Are you writing for a particular type of article, say a very focused, expert-related topic? Suppose I'm writing a topic on a particular receptor in the brain in a particular region. It's a very focused area, right? You need an expert in that area to review your paper, and those are the people that are going to read your paper. Or is it for a wider audience? Maybe I'm writing about obesity. Then I have options of going to multiple journals. But if I'm very focused on some aspect of neuroscience, then those are the journals I'm going to select. So you start off thinking about who is your audience. So with that, you decide which specific journals are you going to write. And that will determine the style of your paper. So if I'm going to write to a neuroscience journal, I'm going to structure my paper according to experts in neuroscience. But if I'm writing for obesity paper, which has a wider audience, I'm going to make it a little more wider, not so specific, because obesity is associated with diabetes, hypertension, so you are broadening your audience. So first of all, you have to decide which topic, which journal that you are aiming to write your manuscript. What movie? What's okay? Okay, yeah. So once you understand, then it becomes very easy to write your paper. So the first hurdle you get through is what type, which audience, and which journal. So 3 things you decide before you start writing your manuscript. And once you have decided that, what do you want your audience to get out of your paper? What is your goal? Are you going to say that I got a cure for diabetes? No, you certainly don't, right? So what is your end goal? What are you trying to give a message in your paper? What do you want your audience to get from that? So if I'm thinking about, say, I'll give an example of yesterday, right? I'm thinking about what is it that causes an offspring to become obese if the mother has a high-fat diet, right? So what message do I want to give to the audience? It's the high fat, and I'm going to end a story with high fat. Yes? So this is how you think before you start writing a manuscript. Any questions? Please feel free, because I won't know whether you understood or I'm just talking to myself, okay? Okay, so please. Okay. Okay, they're following me, okay. When I see you sleeping, I'll stop, okay? So just very briefly, there is a difference between a review paper that you write which you will eventually do, and a manuscript. A review paper is balanced and very unbiased because you are reporting other people's work. You are an expert in, say, fatty acids, and you are looking to see what people have done on fatty acids. So you can do a critique on writing the review paper. I can say, you know, this people found that if they give omega-3, it's good for you. Another paper found that if you give omega-3, it's very bad for you. And so who is right, who is wrong? You look at the methods. You look at how did they do it? And then you critique. You say maybe the difference was because of the methods they used. Maybe the difference was because of the age of the population. So review article is very critical and unbiased in reporting what's out there. Unlike a manuscript that you write is your own work, right? It's all about your experiments, what you have done, and how you're going to report it. Those are the two differences between a review paper and a manuscript. Okay. And in either case, it's very important that you be honest. It's important ethically that you report your experiments as is. Sometimes your experiments don't work out the way you want them to, right? You have this great hypothesis, and you write, your hypothesis was totally wrong. But that's not the end of the story, because your wrong hypothesis may lead you to new avenues and new discoveries. So don't be afraid of negative results. So manuscript is also like telling a story. All of you have read books, right? And you like certain authors because of their style or the way they say or the topic. And one thing you will find in an interesting story book that you read is if they introduce things in perspective, right? If the character is the hero, he comes first, he's introduced first. If there is a villain in the story, he gets introduced as his role comes in. But if I were to introduce the hero at last, you'd be totally lost, right? You wouldn't know what the author or the story is all about. Same thing with a manuscript. You have to lay the sequence of what you are telling. So you have to tell it like a story. If I'm writing on fatty acids, I have to introduce why is it important, what is known, okay? So you have to take a reader through the steps, just like you're telling a story. And like I said, if you walk them from beginning of the story till the end of the story and make it interesting. If I read a book where it says, oh, John came to school, John had his lunch, John went for a break, John did, I would give up the book, right? It's not interesting. Same with the manuscript. Remember, audience is always looking for things that interest them. And even if it's interesting but you can't understand, no one is going to read your paper, okay? So you have to make it flow like a story and make it interesting. And it has to be very concise. You will realize that manuscripts, journals have a page limit, word limit. So you have to tell your story within that. And that becomes a little difficult and easier because you have to choose what is important to tell in your story. Maybe I don't need to tell about the hero's sister in my story. I'll take it out because I have a word limit, right? So you have to make your manuscript very precise to the point. And don't be like, let the reader, like, some of those mystery books, have you read, where they let you guess what the ending is? And I find it very frustrating because it's asking me, after reading all these pages, you figure out who killed this guy. It's very frustrating, right? So don't leave any questions like for the reader to say, what is this paper? What is it asking? What is it telling? And then you say, oh, who's this author? I'm not going to read this author's papers again, right? Because it's so vague, right? So this is how, just think of you as an author, right? There are some authors that you'll read over and over again, and some authors don't inspire you at all. So same with the manuscript, unfortunately, right? You have to tell a compelling story. So what, where, and when to publish. Publish only when you have something to tell. And try to reach as many readers as you can. And one of the things that we consider is the impact factor of a journal. I don't know if many of you know what impact factor is. Yes, you do. Okay, great. Okay, so we all aim for Nature, to get an article in Nature. I remember as a PhD student, I thought, hey, if I get a paper in Nature, it hasn't happened yet, but I'm trying. But those are obviously, the higher the impact factor, better it is for you, right? So just to divert a little bit, we submit grants for funding, right? And then I'm one of the reviewers of people who submit grants. And in that, there is a section where they provide you their CV or bio sketch. And one of the things we always look as a grant reviewer is how many papers have they published and which journals? And one of the comments we give back is he has only 2 publications, but in a very high impact factor, versus I have 15 papers in low. So it's always good science, good journals, makes a lot of difference to a career. Excuse me. So for a good paper, think of this. It has to be original and novel, right? It has to be different from what already is out there in the literature. It has to be comprehensive, means whatever you are discussing has to be, as far as you can to make a complete story. You should have good data analysis, and you should have a good discussion, okay? And I'll take you through this individually. Anytime you want questions or you want me to stop, tell me, okay? So.
Not really a question, but thinking about grant and all that, because it was kind of like that too. We always look at how many articles in the scientific life people. What's your opinion on, like, what would be best? Publishing, like, articles in lower,
Low impact.
lower IF, but
A balance. I'll say a compromise. I always believe in a compromise. Okay, so the way I would say has worked for me is I aim for two, if I can, at least one article in a good high impact journal per year. I have other, like I'm doing an experiment, I have other things that I get out of the results. I know that it's not that exciting, but I would like to report it, and I go for some average journals. So this way I keep up my publication number and try to get into a high impact journal. So the good stories I leave for high impact journals. Does that kind of, yeah.
When you say high impact factors, are you saying above 5?
Yes, I mean, usually, obviously when you start off initially, you know, if I don't have anything great to report, the lowest journal I have reported has had an impact factor of about 2.8, but I always aim for, since then, like average 4.5 or somewhere there, and then for high is always above 6 or 7 in that category. Obviously, not everyone can publish in Nature, which has 30, but if you get a paper in Nature, you made it. Okay? So then I'll be sitting there and you can stand here and tell me about how you did it. Okay, so it's, yeah, so as you go along, right, when you're starting out young, you need publication numbers, okay? Unfortunately, that determines your productivity. So I wouldn't discourage you from publishing in a low impact at the starting of your career, because productivity is what they're going to look for. Because they don't know that you were there in the lab every day carrying out experiments. They only know your name when you publish what you did. And if you don't publish, no one knows. So you have to have productivity at the early stage of your career. At my stage, I can be a little choosy, but initially I did have, you know, the reason why I ended up with Michael was just at the end of my PhD, I had 11 publications. So that is what determines that, oh, she's a very productive student, things like that. So you have to publish, okay? I don't know if you know this saying, publish or perish. Yes, that's true for scientists. Fortunately or unfortunately, I can't decide. A scientist has many roles, right? I told you. We are creative thinkers, we discover things, we design experiments. At the end, we have to be a writer too, okay? So it's a combination, and if you are here, obviously, you are passionate about your work, okay? So.
Also work with a team. We can also balance
Oh, yes.
productive and high-level publications.
Yeah, so Kali has made a good point, yeah, that it's not that I got 11 publications as far where you helped a little bit and managed to get your name as a co-author. Okay, so suppose Maria doing this setting up animals, right? And someone comes and takes, say, placenta or heart or that. She gets to be a co-author on the paper. Automatically, right? So this is how you set up collaboration and work as a team, and you'll get far. I think I can divert a little to tell them. So this is not anything to do with this, but a teamwork. When I did my master's course, I was talking about this to Adriana and them yesterday. I did it in India. And one of the things, at that time, we were told we didn't have PubMed, and no computers were allowed. This course I did was really very stringent. Even for statistics, they didn't allow us to use computers or calculators. Everything they wanted, they wanted us to understand the formula of each statistics. And so we used to have tests, and we were thinking how every other day we had tests. Those courses started at 7 in the morning, ended at 9 p.m., and the next day we had an exam. How do we cope? Especially when you don't have things that you can read on your computer, you have to stay in the library and make notes. So we used to give out topics to each student. Okay, you're going to do mitochondria, you're going to do something on circulation, you read everything, and we used to use, those carbon papers, and write notes, right, and distribute. Guess what we did? We worked as a team, and we all passed as a team. Okay, so if you help each other, I think you would make great strides in research than trying to work in isolation. Okay, so that's one of the key things that Kelly pointed out, that you could get lots of co-author papers as a team. And maybe, you might also find something interesting in that collaboration. So, okay, getting back to, sorry. Okay. So there is good science paper, like I said, and good science reporting, okay? So, like I said, in writing, there are no shortcuts, and practice makes perfect. Like you play soccer, Paloma, every day, practice. The same thing with writing. You plan, you draft, you reread, you revise, and you edit. Okay? Yeah, that's the key to, it's not magic. And what you have to learn and how to improve it, importantly, critique yourself. Okay, self-review, peer review, send it to your colleague, send it to your mentor, send it to experts in the field, get a feedback, and you'll find that you'll keep improving. So this is, everyone knows what bullseye is? Bullseye, dots? Yeah, so you see this blue and white and red circle. So when your first draft is like, aim, it doesn't, right? It's very wide. That's your first draft. Okay? Then you revise, you're getting better. Okay? And that's your final draft. You hit the bullseye, okay? So this is how it works. Okay, so this is kind of a general overview, and I'll try and go through everything. If you find it too boring, just stop me, okay? Because I'm not sure, but you start off, like I said, with a plan. And then you do research, collect, structure your paper, like I said. I'm going to go through this, write it, revise it, edit it, and then, get your mentors to test it, send it out to journals, They will review it, they will reject it, they will accept it, they will ask you to revise it, and finally you publish. Yes? Okay, so, and that's the circle. So, like you said, many of you have published. This is the format of a scientific paper. Title, abstract, keywords, introduction, methods. And I don't know about you, but when I first wrote my first paper, I'm like, where do I start? I don't know if you had that feeling, but hey, I have to do all that? How do I even begin, okay? So these are just some of the suggestions and kind of key points, but I'm going to go through this individually. So title is very important, and I'll tell you why. And title keywords are very important. Also authors. You will always, if you are working in a particular area of diabetes, you might be looking for particular scientist's paper all the time because you have read it, you know they are doing great work. So things like that, so very important. Then I'll talk about this kind is the general order of how you start off writing. Usually title, okay, let's see. Keywords, authors, very easy. But title may not be so easy. I'll show you in a minute. Then you start off with methods and results. Usually that's how I do it because it's very easy, right? You know your experiment, you know what you've done, and you know what results you want to report in that paper, okay? Once you do that, then because you have got your results, you know what your introduction should be about and how to focus your introduction. And then comes the difficult discussion part, okay? Okay, so I will. And this is basic concept of a manuscript. You've seen this kind of a funnel thing, right? You start broad at the top. So introduction, you start very broad, and you focus down to your hypothesis, which is very narrow. Your methods and results are very narrow because you are not reporting on methods that you haven't done. So that's very focused. You are only reporting on your results. That is very focused. And the discussion is very focused on your results and broadens out in context with everything else. Okay, so this is kind of the structure of your paper. So now I'm getting actually into what you said, nuts and bolts of, like, you know, when you architecture design the building and then the construction actually starts. That's what I'm going to. Anyone wants to take a break before I do this? Are you tired? Okay. Cover letter. Yes, I will talk, or I can talk a little bit now. Okay, so cover letter is mostly to a journal. It says, dear editor, or whatever journal you're writing. The title goes in the cover letter, who your authors are. And you have some journals nowadays, in fact, most journals require you to tell you them in few sentences why your work should be considered for publication in that journal. And what I find most, I would take my the importance. So can I just write somewhere on the board because I don't have that slide? Yeah. Okay. So on the cover paper, your significance. receives thousands of manuscripts. Okay? So your sentence, so this paragraph, this is all in few sentences, okay? First sentence, what you're talking about? So if I'm talking about obesity and fatty acids, maternal obesity causes offspring obesity likely through fatty acid pathway, okay? I'm just making this up, okay? So you said our results show. What is your key finding? Okay? That comes in the second. And how is it overall relevant? Very few sentences, you have to tell this in your cover letter to grab an editor's attention. So once you do that, that's all, because remember, editor doesn't want to read your whole abstract. The abstract is there for the editor to read. The editor wants to know what it is and how he can. So suppose if I was talking about obesity and say fatty acids. Here my significance is maternal. That's my subject, right? Maternal, say high fat obesity, right? And that's my subject. The outcome is offspring. Right? Whatever it is. So, and my results show something, that one particular fatty acid is the driver of this. Okay? And this is causes, say, increased body weight, increased food intake, whatever. And this could be fatty acid mediated programmed obesity. Okay, I get the gist that what your paper is about without reading the whole thing. So the cover letter is also very important. And nowadays, more and more journals require you to summarize. So this is your, and then they ask you something, I think, about no conflict, and that you have, so if suppose you are testing out a drug, and you have got funding from the company that has provided the drug, you have to declare that in your cover letter, whether there is a conflict. Because if you are reporting a drug that's provided by the company, and the company is funding the research, that's conflict. That has to be declared in your cover letter, okay? So those are the key things that you have to do. And that everything you've done, if you're doing human research, it's ethical. So that's what a cover letter entails. Okay.
Mina, I want to give an advice for our students, because all the
You know what I mean?
Being attractive, and fair, because you have these results and all the literature said the opposite. And then you get this, obesity, not causing inflammation. How to tell this to the readers, but being attractive and being fair with your results.
Okay, so can you want me to address that? How to say, like how to present negative results?
An advice.
Okay.
What to
Okay.
do in
So here is what I would do. if I had negative results that was contrary to everything that was published, first thing I would do is repeat it. Okay? Because I'm going against everything that everyone has said, so I better be armed and prove my point. Okay? So I want to be well prepared to defend myself. If I'm going to say, hey, you all guys are wrong, I'm right. So, yes, because I don't want to be slaughtered out there, right? So, what you need to do is repeat. And if you are convinced, don't be discouraged to stand up for it or to report it. Because that's how things change. That's how science evolves. Remember, the technology that was available even 5 years ago is different today. We have better technology. So I remember again, I go back to days because it's in context of time. When I was looking at the liver and that, a certain receptor hadn't been discovered by then. So people didn't understand the pathway. And they speculated and they did that till someone discovered the receptor, and everything we thought about was wrong, right? Because we didn't know this receptor existed in the liver. Okay, so don't be afraid for a negative results. So again, I was doing this experiment as a PhD student on protein restriction during pregnancy and lactation. And just before I left, I was looking at the aging, right? How long do they live? And I thought, this low protein during pregnancy and lactation has all this insulin secretion problems, blah, blah, blah. It will have the least lifespan. Guess what? It lived the longest, even compared to the control. And I'm like, this is not. And right now, what do you say? Caloric restriction increases lifespan. Okay? It's all, and the telomeres and everything has come out then, right? So at that time, I didn't have time to repeat it, so we buried that result in a review paper. But I was not wrong, right? So what I'm saying is that sometimes don't think that a negative result is terrible. You never know, in future someone will say, hey, that person who reported this made a discovery that no one was aware of at that time. And it's happened. I think you can vouch for it, right, Márcio, Adriana? That we recognize discoveries that this discovery was made long ago that no one had paid attention at that time. So very important, like I said, verify your negative results. Okay, and think critically. So suppose I say that I did not see inflammation in a certain area of I'm taking again the example. We didn't see it in the hypothalamus. Question I would ask is, I'm looking at feeding. Do I need to go more specific to a specific region? Will I see a difference? Or if I challenge, will I see a difference? Okay, if I answer all those questions and I'm still stuck that there is no inflammation, then you have a story to tell. Okay, so negative results is not sometimes the end of the world. Okay? Okay. Okay, so now actually we are getting to how to write a manuscript. So I always start with an outline. And I'll tell you why you need it. It gives your paper logic. You start thinking logically, and it gives you a structure and an outline to follow, right? I don't know if I'll give you a simple example. My sister, when she goes grocery shopping, she has a list in front of her. She goes into that, bum, 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 and she's done. I go to the grocery store, I go to the aisle, I go to another aisle, and then I come back, oh, I forgot that. I forgot this. And I'm all over the place, right? So I wish I had learned my sister's way of doing it, right? Because it saves time, it's efficient. So same way with the paper. It requires, and I'll show you what an outline looks like. It gives you a structure to your manuscript. It gives you also what is essential for you to include in your paper. And it helps organize everything, all your details, okay? Remember, you have worked on your experiments for maybe a year, 2 years, and you are going to condense all that work in 7,000 words. It's not easy, but that's what you're going to do. Okay, so this is outline, title, abstract, introduction, methods, results, discussion, summary, and other stuff, acknowledgements and that. So I'll come to the title in a bit, but an abstract, under abstracts, you're going to write aims, methods, and your key findings, okay? That's what your abstract is about. An introduction, you write, begin with why your topic is important. Then you write what is already known in literature, what is missing. What is your research objectives, and how it addresses the knowledge in gap. That's very important, okay? And then brief results with your conclusions. That's what your introduction consists of. And then in the methods, you write the site description, experimental design, analysis, both biological and statistics. And in results, you describe experiment 1, 2, and 3, and so forth. And your discussion, your opening paragraph, and I'll go through all of this individually. You have summary of your key results, and you then go systematically discussing each of your results. And then overall, connecting your current study to what is out there and explain what you have, how you have breached that gap, okay? And then summary and conclusion, again, main findings, significance, how is it applicable, and what is future directions. So in a nut, that's how your paper, so if you have this outline, that's how I'm going to work. So this is what your outline looks like. Okay, now just think of title and abstract and keywords. Believe it or not, these are the most important part of your manuscript. Because if I were to go to any of the search engines, PubMed, I'm going to look for your title, keywords. If you use keywords that are not relevant, I'm never going to find your article, okay? So very important to use the right keywords and the right title and an abstract, because those are your selling points. It's just like advertising, right? Any products that are not advertised well never get sold. Same with your manuscript. If you do not have this, it's not many people are going to read your article. You know that. Another thing they talk about is citations. How many citations did your manuscript get? Apart from the impact factor, people are interested now in what they call as H index. I don't know if you know how many times your article has been cited. Okay, so very important that people find your article. So, like I said, use the right words and give the best chance of your paper being read. So, have you scratched your head and said, what should the title of my manuscript be? Okay, this is what I follow. You have the research question, and this is the answer I'm going. Okay, so you ask yourself, what is my paper about? So this is as an example, okay? My paper studies how program volume affects outcomes of liver transplant patients in waiting lists, okay? So what methods and techniques did I use to perform? It was a case study. What or who was my subject of the study? I studied 60 cases of liver transplant patients on a waiting list throughout the US, aged this. What did I find? My study revealed a positive correlation between wait list volume and negative prognosis of transplant procedure. Okay? These are my questions. This is what I've answered. So then, in one sentence, this is the answer. And so my paper studies how program volume affects outcomes for liver transplant patients on waiting list. What are the keywords here? Program volume, liver transplant, waiting lists, and outcomes. Okay? This is a case study, so the keyword is case study. I studied 60 cases in a transplant patients waiting list, throughout USA, 20 to 50 years. It's USA, age 20 to 50, 60 cases. And again, my study revealed a positive correlation between wait list, volume, and negative prognosis of transplant. So positive correlation between wait list, volume, and negative outcomes. Okay? So here you have your answer keywords. Oh, I have to do that? Okay. Okay, so now I have a title. Okay, I've sorted out what my questions are, what my keywords are, and I've got title. I said, we employed a case study of 60 liver transplant patients around the USA, aged 20 to 50 years, to assess how waiting list volume affects the outcomes of liver transplantation in patients. Results indicate a positive correlation between increased waiting list volume and negative. This is too long, okay? So what do you do? But I have the gist of what I want to say in my title. Now I'll start trimming it, okay? So what I'm going to do is I'm going to remove elements that make it a complete sentence. I don't use there is or was. That's not required in a title. And I'm going to delete all necessary or redundant words that are not central to the study. And I'm not going to use database search, okay? So here is the original title. I've deleted we employed. I've deleted all these articles in patients, results indicate after transplant procedure, okay? And I'll shift some words around and rephrase it. And now I have a shorter title of a case study of 60 liver transplant, blah, 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 and I've come down to 38. Still not good enough. Okay? So the number of patients I studied and the exact outcome is not relevant in a title. So I'm going to remove that. So I come up with, which is shorter, with 19 words, you can read it, but still too long. So methods is, the methods used in a study are not mostly searched for in keywords. I don't look for Western blot in my title or that kind of thing, so I'm going to trim it more. So I've come, after deleting all that, I've come assessing the impact of waiting list volume on outcome and prognosis in liver transplantation patients. See how concise it became? But I started with everything I had, what I wanted to say, and I've trimmed it now to 15 words. Okay? So you'll see from the title, the final version, you can see the subject and the objective of the study, aims, and what you want to achieve in the title. So where did I start off with a very boring, very long title, but it had the key elements that allowed me to trim and focus it and make it more attractive title. Okay? Any questions?
I have a question about the methods you were thinking that in the title.
Yeah.
Because some trials of trials and some studies we use to start. I think it.
She again raises a very good point because I'm coming to.
We have to.
Yes, and this is the next slide.
Okay.
Yes, so you can add a subtitle, which is what Kelly was asking. So what I've done is add a semicolon and said it's a case study of patients. You can do that. Okay, so I might write it depending on your choice. I could leave it, but if it's required, I could add a case study of. Yes? Study? Yeah. Yes, yes, absolutely. Okay. No, absolutely. I mean, you might have something like a title which says, okay, here, maybe I can do this. Okay, so suppose I have a title of effect of fatty acids, okay, effect of, hold on, maternal HF on offspring obesity. Doesn't tell me much, right? It doesn't say anything to me, right? I could then say maternal high-fat diet mediated, and I can have an outcome. That tells me more than the first title. So you are right that if you, can you include? Yes.
I heard from a colleague, she said we are having this first time working together. She's doctorate too, but from other area in university, and she said sometimes I don't like to give the answer, give the results there because people could be interesting to get the whole paper to read.
I think it's choice, but like I said, your title, keywords, abstract is the selling point. If I don't catch that on my Google search engines, I will never find the article. Okay. And I'm sure all of you have used PubMed. Have you struggled finding the right keywords to pull out an article? Yes. I get all the time. I'm like, which is the word? I'm sure that someone has published something in this area. Why can't I get an article on this, right? So using the keywords is so important. So I would say it's just like, hey, let me take an example like everyone knows designer clothes, right? Gap, Gucci, this thing. Why is it? Because they're advertised. Do I know a small dressmaker in the corner of a street who makes fantastic dresses? No, because I've never heard of him. And I could get a bargain there, but I haven't heard of it. I will never shop there. Same with writing a paper. I'm sorry, but it's all about marketing and getting your paper out there.
And do you think the title is better to give the results also, or
Just the outcome, not the entire
give
results.
some tips
Yes.
what they will
Yes.
expect in
Yeah.
the
You could say here, maternal hyphen mediated offspring hyperphagia, right? It's not giving out the entire, but it's telling you the outcome here.
more interesting.
Yes, yes, yes. So,
And how about the place? You said there, say, for example, Brazil or São Paulo.
yes,
or
yep, yes. You don't have to be like specific like Los Angeles. I can say it's a US population. Okay? Also, for animal work, most of journals require nowadays to tell whether it's in rats or it's in a primate. So you might, instead of a case study, you could say pregnancy during rat pregnancy or something like that. But people, more and more journals require to specify in the title whether you're working with animals or humans.
Nowadays,
Anyone wants a short break before I plow on? What do you think, Adriana?
yes, I believe that we can have a break now
Okay.
to go to the restroom and right.
Before everyone
Sleep.
Okay. So I will finish with the title, and then Callie asked about authors, and I'll address that. But to summarize, a good title, the headline has the main result, not the effort. So it's not the experiment that I did, cell culture study, you can say, but what was your main result in that? Paper has to here. So you begin with a keyword for target. So if I'm writing about, say, obesity, I would start with, that's my audience. And what's specific to your paper? Say, it's fatty acids, okay? So target audience is obesity, fatty acids, accurate picture of the article, motivate the reader to go and read your article. The title will be the one to tell them whether I should read this paper or not. Keep it short, but like I said, the first one, which didn't convey any meaning, is not going to help either. And don't use question mark. Don't write your titles as a question. Never, okay? And you don't have to write complete sentences like I said, there was, or something like that. You don't need that in the title. And be realistic. Don't say, my drug X cured diabetes. It didn't, okay? So don't overstate. And like I said, you always mention whether it's a clinical or an animal study. Okay, now I'll go briefly here. There's Kelly asked experiments. You get to be the first author. They are the senior authors because they counsel you, they provide ideas, they bring in the funding. Okay? So if your first paper, maybe no one knows you in the literature world, but they know who Torsoni is. Okay? So your paper will get picked up based on your mentor till you establish yourself. Okay, this is how, unfortunately, the literature world works. Okay? So suppose you have two students that did equal work on that project. How do you decide who the first author is? Now, journals allow you, say this is Smith here, and this is Jones. You put an asterisk here and said equal contribution as first authors. Okay, so both Smith and Jones are first authors. And then you have other people that have helped you. Say you are working on a placenta, and someone helped you process or section the placenta, staining, and read and gave you the results. That merits a co-authorship. Suppose someone provided you a cell line that you used. That doesn't necessarily constitute co-authorship unless you had an agreement. You can put that in acknowledgement. Okay, so it depends on what agreement you made with this person. You would, if you had agreed that you just provide the cells and I'll put you as a co-author, then you have to do that. Okay? Otherwise, you can put that cell lines were provided by. That goes in the acknowledgement. Like in my lab, I have technical people helping with the essays. They don't get authorship because our service, like I pay them to do the work, they go in the acknowledgement. Like I say, Stacy Jones helped with also the animals and that, and you acknowledge in the acknowledgements. But anyone who has contributed somewhat substantially to the paper can be a co-author. But usually the credit usually goes, this is important, not that this is not important and this one is important. So suppose Adriana is the main author, and it's very rare that Adriana will put herself as the first author, because she's reached a stage where this is more important than this. Okay? Because as a first author, you are still doing the actual work. The senior author is running the lab. That's the difference, okay? Does that address? Yeah. Okay. 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 So now we go to the next stage of writing an abstract. This is the next most read part of the paper, as I said. It should be easily accessible on PubMed, and it summarizes the whole paper in a very condensed message. And this will only be read if someone thinks your title is worth reading. Okay? Title, very, very important. Okay, this is a little dense, but what does an abstract include? It has a brief introduction that says what is the subject. And it's always one or two introductory sentences. Abstracts have a word limit. There are some journals will give you only 150 words. Some will be 250 words. It depends which journal you are publishing in. So you have to be very precise, okay? In that, you have to explain your topic, the purpose, and the research question. If the background is needed, but it should not be more in depth, the main thing of your abstract, okay? Material methods is very, again, one or two sentences. How did you conduct the research to generate the results? And you may include what type of statistical analysis you did or data. Simple thing you could say, we did protein expression and unpaired t-test or something like that, but very short. You don't have to describe protein expression by Western blot. Okay, the result, why is this important? What are your discoveries? And you can, this is, like I said, the bulk of your abstract. You actually condense your results here. And discussion, conclusion of future directions. What is the take-home message of your paper? What did you find at the end? Did I discover that the fatty acids causes increased food intake? If I have found that, that's my take-home message. Then you write one or two sentences containing your conclusions and recommendations. And your conclusions are the study's contribution to the field. What does that mean? You remember I said, what gap of knowledge are you trying to achieve? And when you conclude, you say, or whatever it is, showed that omega-3 is very critical for cognitive development, okay? That's what you have established. So that's how you end, okay? What is your key finding? So this is basically the abstract, not very difficult to write, right? Everyone agreed it's not difficult to write. I was like, okay, that's great. Yeah, it is difficult, but usually what I do, I write my abstract the last. Okay, because I have the framework and now I can extract. So abstract always goes last for me. Okay, remember introductions, we said start big and narrow down, right? You have to keep it relevant and stay on the topic. if you are talking about, again, sorry, I'm using your example of yesterday's. If I'm talking about fatty acids, that's my story. I'm not going to be talking about, say, other hormones or cytokines, because for me, to find a pathway that connects it and tell a story is going to be very difficult. And remember, if I can't convince myself, I'm not going to convince you, right? So I have to be convinced that this pathway stands and I can make a case for it. Because otherwise, like I said, no one reads it, okay? You establish your rationale, significance, and hypothesis. What is rationale, right? Why are you doing it? What was the reason behind? Why was it important? And what is your hypothesis, right? People, you know what hypothesis is, right? Mm-hmm. So those are the key things in your introduction. This is the big thing. The literature and all that, this is the narrowing part, because that's focus. That's focus particularly to your paper. Okay? So we'll ask a few questions before you start doing the introduction. These are the questions. What is known in the field and what is lacking? What gaps does your study address? What behavior or results did you expect to see? And how was this behavior examined? Those are the key things that you address in your introduction. So step one, like I said, introduce your topic, describe the background, establish the research problem, and specify your objectives. So again, this is repeating the same thing that I said in the previous slide, but I'm just putting it as step 1, 2, 3, 4, okay? Okay, so this, how do you start? So I've told you what absolutely your whole introduction is about. What is your opening paragraph of your introduction? Okay, it should provide the reader with indication what is to come. And I'll explain what no content statement, cliches, and plagiarizing means, okay? So you have here, since the past 2 decades, scientists have been interested in obesity and life expectancy. Really? Doesn't make sense. And you don't make claims like that imply that you are going to solve the lack of knowledge. This sentence, obesity has been investigated extensively as a model for life expectancy in general. Or despite intensive study, little is known about obesity. Wrong statements, right? Overstating it. Yeah. So you avoid all this kind of cliches, what we say cliches, okay? If you want to set the scene and grab the reader's attention, obesity is known to be associated with reduced life expectancy. The mechanism is multifactorial with interaction between genetic, epigenetic, and environment. Now I get a clear picture, okay? Before it was like, huh? Really? Okay, so this is how you start in few sentences, but grab. I know I go back to a novel, but I find that if first few pages don't grab my interest, I don't read the whole book. Same with your paper, okay? Okay. So then you come to the middle of the introduction. And one thing is clear. How many of you, you've read many articles, how many of you really read line by line of the whole article. There, right? So really, a reader is not going to go through every line of your that. So in the middle, very few people will read your entire paper. So like I said, the way you write is the journal format will help you guide your introduction. And another thing you find is what it says, the sign points or subheadings. In introduction, you may not use it, but in results and this kind of thing, it helps you clarify, okay? Okay, so if I'm talking about obesity, fatty acids, appetite, start with the broad thing, how obesity is. Fatty acids, where do they come from? Okay, what is it important for? How does it relate to appetite? I'm connecting now the dots. And what is my hypothesis? And what was my key finding that you state in introduction, okay? Okay, so any questions on introduction? Before I move to material and methods? No? Okay. I don't know if you guys can see this clearly, but this is important. Remember, this part serves for people that, okay. This is important if I am interested in replicating your study. Okay, and it's always good that people can replicate your studies. So one of the things that happened, and this is again I'm giving an example of NIH, the biggest funding agency in USA. It's called National Institute of Health, and they give out a lot of funding. We scientists depend on it, literally. There are other agencies, but this one is like the federal that we get the money from. One thing they found out that after giving this funding to so many investigators, complaints came back that some of the things cannot be reproduced. And this is funded by NIH. So what has NIH done is when we apply for grant, they want a section that says authentication of biological things. You've done it for our grant. What it means is they want you to detail where your source of antibodies are coming from. Because you could have in-house antibodies that for some reason only work for you and doesn't work for anyone else, okay? They want to see whether your results can be reproduced. Okay, so it's very important that when you write materials and methods, you give enough details for anyone who wants to reproduce your results can do so. And it goes to your credit because if, so this Dohart field, when Barker first published his work, that he showed an association between, so let me step back, okay? He comes from United Kingdom. And there are regions in United Kingdom, just like any other country, that have socioeconomic differences. So there's a region in UK that's poor and has poor nutrition. So he saw that first observation he made was there was increased rate of cardiovascular mortality in this poor region. And somehow by chance of his thinking, he went and looked at the birth weight records of this population and found them to be low birth weight. And he came up with this Barker hypothesis, which is now the Dohart field, where he associated low birth weight with increased risk of adult cardiovascular disease. So at that time, people were skeptical because they said it had to do with socioeconomic status, nothing to do with nutrition. And many people said this is not valid. Guess what? 10 years later, everyone, not all studies, majority of studies around the world have been able to reproduce these findings, which now gives credence to his hypothesis that adult disease will originate early in life. So reproducibility is all about giving credibility to your studies, okay? So step 1, remember your method section serves the purpose, helping readers understand the study and helping them to replicate. It should provide, like I said, enough information for them to replicate your work. I think I'll go back. Sorry. And step 3 is you have to put yourself in that position. What details do I need to replicate the study? If suppose you were using a new method or a new cell line, you should give sufficient details about the cell lines, okay, including, if you were comparing 2 groups, how many cells did you see? Well, there's the same numbers for both the experiment. So those kind of details is very important. And step 4 is consider this, if you have complex study design, you might want a flow chart that explains the study design better. And then, okay, you have to adhere to all the ethics. For people who have done human studies, you have an opening paragraph where you said ethical approval was obtained. This is the IRB protocol number. If you're doing animal studies, you have to do the same. So those are the ethical questions that you need to address. This was one instance that we were doing this study, doing a drug testing on the mice, and we were approved to carry the mice only till, I think, 10 weeks. And then I had some health issues. The mice went to 12 weeks, and guess what happened? I could never publish that study because I did not have the permission to take the mice till 12 weeks. Okay, so very important that you be honest about the ethical issues, be it human or animal, okay? So you always see materials and methods. I'm sure you know, I don't have to tell you this, but material refers generally to human, animals, tissue cells, what intervention drugs or devices you use, if it's anything to do with instruments and so forth. And methods actually refers how the experiment was done. Okay. So basically, I think this is no-brainer for you, so I won't spend too much time. It's just that the subsection, this is your study design. So if it's clinical, you have the ethical. What was your selection criteria, your methods? Was it prospective or retrospective? Was it interventional, so all those details are provided in materials and methods. If it's animals, you need all this description. Cell culture, you need all this type of. And then you have how was the data sample analyzed? If it's, sometimes everyone does Western blot. I never describe my Western blot. But what I do is provide my antibodies, source of antibodies, the dilution that I did, and the KD. But I don't say, oh, I did this, I did electrophoresis, then I did. I don't do that. Same for RT-PCR. You provide the prior sequence. Everyone knows what it is. But if I were to do some novel technique, then I would provide details. Okay, and obviously this is very important too. Many papers get hammered because of use of wrong statistics. So be sure that you use the right statistics and report. Because, I don't know, as a reviewer, if you see wrong statistics being used, I doubt your whole paper. Okay? Okay. Any questions there? We're coming to an end. I know you guys are tired. Okay. So, result, structure, and approach. This is very important, a good structure. I don't know, but I have read papers when I have to go back and forth between the figure and the text, and I get very frustrated. It's very bad when a reviewer gets frustrated. Be sure he or she is going to reject your paper, okay? So, don't, say this increased, and you look at the bar graph and you say, what? And don't use the fact that it was, if it's not significant, it's not significant. Don't try to make a case of saying it was slightly don't do that. In the discussion, you can say there was a trend, okay? But in results, if you are, if you are using tables and bars, don't keep repeating the results. You've given a bar graph. I can see on the bar graph. I don't have to write means and standard error in brackets of everything. Nothing is more frustrating to a reviewer. Choose, do you want to report your results as mean and whatever standard deviation in the text? Then go do away with the bar graph. Don't have both in it, okay? And if you are very fastidious and you want to include, put the mean value on the bar. But don't do both. Because to read through all those means and standard errors and then look at the bar, and I'm like, it's not, going to help your case at all. So there are two ways of presenting and organizing the results. Okay, sometimes it works, sometimes it doesn't work. Okay. Oh, sorry, I've done both of them, it's okay. So this is an example. This is you may have noticed, this is you might report unusual correlation between two variables during the analysis of your findings. It is correct to point that out in the result. However, speculating why this happens and offering a hypothesis, all this goes in the discussion. Okay, results is what I found, period. But you can use, like I said, suppose I see an increase in one gene, and I expected the outcome to be related to the increased gene, but it wasn't. You can say paradoxically or in contrast to what was reported, but you don't speculate or offer a reason in the results why it happened, okay? Some papers, having said that, it all depends on the type of journal. Some journals you've seen, they report results and do the discussion same. There are some journals that want it that way. So in there you can have the discussion, and you always provide a brief conclusion at the end of that one results section, okay? So it depends on which journal you are aiming for. But overall, most of the journals prefer results, and discussion. So you start with, suppose I'm reporting one results, right? So the introductory, for understanding, you restate the research problem that you are, I'm looking at gene X, right? So you start off gene X, which is related, which is the key marker for, say, appetite or something like that, or for satiety or something like that. You start off and you say what was it, and you arrange them sequentially. So it's very important that you have appropriate data analysis and at least interpretation of your results. Okay, I cannot imagine if I said that I gave a drug, glucose levels went down, and insulin levels were down, and I say I still refer it as a diabetic state. It's not, okay? So be careful how you interpret your results and showing them in the results section. In your statistical, like I said, significance is very important. Don't, I've seen papers where they say P is less than 0.00001. I'm happy with 001. I don't want 0.000, right? Or I don't want 0.012, right? You use either 0.05, 0.01, or that. And if you are showing a trend which is very close to significance, I'm happy with 0.06 or something like that. But you say it was slightly increased with 0.12. No, it's not. Okay? So, yeah?
I have a question.
Sure.
What do you think about to use tendency? For example, tendency to higher spread, low, for example.
Trend, yeah.
Trend.
Yeah.
Sorry.
Oh, one. It's very difficult to get it to significance after that. Unless you, I don't know, it's really a widespread. So that's the other thing is, this is all about, I'm not talking about it, but you have your data. Okay? If you, that's messing up the results. Things like that. If you I would say, yes, there was a trend. And I could believe it because I see that one outlier. Okay? Okay, where are we? So, yeah. Then the middle, sorry. I think I'm getting, okay. So, like I said, you summarize, and then you have all these figures, blah, blah, blah. Some journals prefer it in the middle of the text nowadays. They've come up with these new ideas where literally you are the editor of the paper you're writing, and you have to plug in your figures in there for them. Before, there was a trend at the end of the paper, right? You attach your figures and tables. So it depends, again, which journals you're going to. And, okay, highlighting. Okay, so here is some of the things that, when you, and I'll come more to the style of writing, but results are always reported in past tense. It's not happening currently. It's already done, okay? And obviously, how much data to include and what type and all that will depend on the length of the journal. Okay? So now let's talk about what to avoid in the results. And I think I have a big list, but maybe I might not get through all of it. But one thing I've said is don't discuss or interpret your results in results section. It's in the discussion. Reporting, again, background information, not in the results. I've seen this happen many times. It's in the introduction. Ignoring, this is what. Okay? Okay, don't ignore negative results. You can weave it through your story that you found this, and you can be honest. You can say, right now you don't know, or it's unknown why this happened, but maybe future studies can address this, okay? But this comes in the discussion part, but then you can include your raw data. So that's the other thing. Every experiment you do does not have to go in that paper. I don't know whether you do, but I do some experiments that I feel doesn't fit the story. And it's not essential that every experiment gets reported in that paper. Okay, it's all about how you're going to tell a story for that paper. And some papers now require raw data. If it's a gene sequence, you have to deposit that. If it's a Western blot, they write to original blots to be deposited. So all that is raw data that some journals require. Okay. And like I said, be concise and factual about what you are. Do not use vague or non-specific terms as appear to be greater or lesser than, or demonstrates promising results, okay? Don't use phrases like those in results sections. And presenting the same data or repeating the same, don't do that, okay? If you need to highlight some of your key findings, do it in the discussion, but don't keep repeating it in the results section. Okay, so, and confusing figures with tables. I've seen this very often. When you are comparing multiple groups, you have A, B, Cs, and it's very difficult to figure out what the person is saying if the text doesn't guide you. So always be crystal clear. Assume the reader doesn't know what you are doing. So explain them. It always helps to explain with clarity than to let me guess. Because if I'm to guess, I might guess wrong, okay? So don't let the reviewer guess what you are thinking. Okay, so now we come to almost final part of the paper. Any questions? No? Okay. This is not much. It's very, the goal is to critically analyze and interpret your findings. You should, so like I said, you open the statements. I always like starting my paragraph with, if I had novel or new findings that suppose about appetite. People know that although it's known that ghrelin, whatever is involved in enhanced appetite, this is the first study to show. Point number 1, what did you show? Point number 2, okay? And then you summarize before. That's the opening statement of your discussion. Always highlight your key findings. Then you, and put now the second and that in context with what is already known. How does it compare? Did anyone, is it consistent with other authors who have worked in this area, or is it different? And so organized, concise summary and critical discussion of your major and minor findings, acknowledgement, acknowledging what is the limitation of the study you have done, and the future direction. Where do you see that your study going next, okay? And a strong ending is very important, which now summarizes the significance, overall conclusion, and major impact of the study. That's your beginning and end are your punchlines, okay? So be very critical, be very concise, and pay attention a lot to that. Yeah?
I always have a short question about how types of limitations we have to discuss in the paper, in the discussion, like just sample size or methods that are not that good for the problem we had. What kind of limitations do we have to acknowledge in the discussion?
Okay, so good question. I don't always acknowledge limitations in all my papers. If my study has clear-cut results and I have, I wanted POMs to go down and NPY to go up, and it happened, fine. But there was a trend which was almost close, then I would say maybe my sample size, you could address that. Or you could say that this was done at one time point. Okay, you don't know, suppose you did it just in the fetal stage. You don't know whether this persists. That's the limitation of the study. So it all depends on the context of, you don't have to report everything, your limitations, if there are none, right? Usually in human studies, that's more critical because you are doing with a specific populations. It can be your size, dropout number, the limitation. We had a lot of dropouts, your small sample size, or we were biased because we focused on particular ethnic groups, or something like that, right? With animal and cell work, that issue doesn't come up, but it's more of methodology. Suppose you used a method, and you had done this work maybe a year ago, and recently this new method came up that sort of not undermines. Well, you say, well, this was future studies would be needed to use this technique to validate this results, that kind of thing.
I always think that the limitation has to be things that you can't control. Like, when I'm using the example of Mayara's study.
Yeah.
We are using fetuses, so we can dissect the hypothalamus and just push the arcuate nucleus. So it's not as impossible, but it's hard. It's really difficult to do. The sample number, it's something that you can increase, right? So
Yes.
I never know if it should be in the limitations or.
No, so here's what I would say with her. Okay, one, where would a sample size? Suppose she had not that it would be acceptable, but I'm just giving an example. She had 4 and 5, 4 in control groups, 5, and she showed 0.08 as significance. Chances are, if she increased to 8 and 8, she might see a difference. That's when I would say. But I don't think any reviewer would accept I would ask them to give me a power analysis of how they came to N is 4 to use in a study, okay? But that's just one example. For the hypothalamus, you could say that this was done in the entire hypothalamus. But if you are reporting also 3 weeks where you have done arcuate and see the same changes, then it's not a limitation, because maybe in fetal, all those lines, the regions are not so well defined. It's still developing, right? But if you're reporting at one time, you say, okay, it's difficult to dissect out the arc at this stage, and these results reflect the entire hypothalamus. Whether this would still, if specific to arc, would probably, if you see it in the whole hypothalamus, I imagine arcuate would give you stronger results. Okay? So. Okay, we're coming to almost the end. Okay, so I'm just summarizing the discussion structure. I don't know if you can read, but I think I've repeated this quite often, so I will just skip this part. It's just provide the information, second paragraph, and summarize. It's just the same thing that I said previously. Okay. Now conclusion. This is the final part, I promise. Then I just a few slides of styles, okay? So basically, you discuss your overall conclusion and the major impact of the study. This part is very important, because what is your main take-home message? What do you want the readers to understand about your research? And what is the main contribution your study made to this field? Relate this section to your first paragraph of the discussion. Remember I said with the opening line, my key findings, right? In other words, how does your study fill the gap or address the problem that you presented in the introduction, restated it in the paragraph 1 of the discussion? So basically, conclusion is like bringing the whole plot together. Okay? And with that, there are some kind of suggestions of writing style and things that I'm happy to skip over if you guys know about it. I don't have to tell you. Do you want to hear? Okay. Okay. So being critical, you need to be critical of the work of others, but more important. I don't know, but Michael sometimes says I'm a pessimist. I'm very critical, but you need to be critical of your own work. If you do that, then you have questioned and answered your own problems before the reviewer raises them, okay? And there is no such thing as scientific fact. It's always based on observation, experiments, and that, right? That's what science is about. It's not, and it's important to identify what is not known. Critical assessment presents enough information to recognize the merits of a particular interpretation. In other words, when you, like, I mean, sorry, you don't mind me keeping So we were looking at the data and we said, okay, why does this happen? Why is, if it's sucrose and you think it's the culprit that's causing inflammation and our control diet has higher sucrose levels, why is the maternal blood glucose high in the high-fat diet? Okay, so how do you do that? One of them came up with a brilliant idea that we're going to use cell lines to test in vitro the effects of sucrose and this omega fatty acids and see what happens to the cytokine secretion. There you were critical of your work and you found a solution to figure out and point out, yeah, it was the sucrose in the diet. So if you are critical, you'll come up with how to address that critique. Okay, so very important to do that. So this is just an example. This is very uncritical. A statement like showed that diabetes could be cured, right? But if showed that diabetes could be cured based on normal glucose levels after being critical is, however, the experiment was flawed by lack of appropriate controls such as non-insulin. That's being critical. Okay, accepting the first statement is not being critical. Okay? Okay, so here are things that you do once you do. Always proofread your manuscript. Make sure you use the same abbreviations and symbols throughout. You can't write TNF and then write one with a hyphen, one without the hyphen. Make sure that you are consistent. That's considered sloppy work by a reviewer, okay? Avoid repetitions when you're writing. Don't write the samples were collected, the samples were stored, the samples were analyzed, and I will go to sleep. Okay? You can write the samples were collected and stored at minus 80 analysis. So don't keep repeating yourself, okay? And use passive and active appropriately. Experiments were carried out. The results show future work will, okay? So use appropriate and avoid long sentence, complex words, and flowery language. We are not poets, okay? So avoid that in science. And do not describe every point of the data. I find that very frustrating. I don't know if you've read papers, but if you have a bar graph that describes group A was slightly increased compared to the control, group B was significantly different from group A versus group C and group D, and by the time after that, I don't know what's happening, okay? So don't describe every data point, okay? And now these are some style suggestions, okay? They're just suggestions. One idea, one sentence at a time. Don't try to cram too many ideas into one sentence. Concise, one sentence is about this. Don't say and because, and your one sentence is like half a paragraph. Don't do that. Okay, and paragraphs, like I said, one topic, one paragraph describe, go to the next paragraph. Always start with a lead sentence and connect topics. So if I'm talking about obesity and how bad the problem is, and the next I'm talking about why it occurs, connect, go to the next paragraph and connect the mechanism for etiology of obesity, and then describe, okay? Transition to the new topic also, do it like that so it flows. So now I'm bringing fatty acids, say, into my third paragraph. Then I will say lipids, particularly saturated fatty acids, are known to be associated with. So introduce connecting, like make it flow, and always end with a summary statement, okay? So these are, like I've already said, clear, precise. Are you talking about mRNA or are you talking about a protein? Nothing is more frustrating than trying to figure out Did you measure mRNA or were you talking about proteins? Okay? Like I said, be forthright. Don't, don't think that you have conquered the space and, kind of statements. It just doesn't fly very well. And jargon is, don't use unscientific terms, right? Just that science is science. And avoid something like interesting to know as a matter of fact. Those things don't go in science papers. And eliminate this, like I said, make sure your paragraph 1 to 2 to 3 flows naturally. It's not like disjointed. All of a sudden, I don't know why you were talking about hepatitis when I have no clue what it is all about, okay? So, connect everything. I think all this I've said. And oh yeah, I'm sure none of you do that, but one thing is don't plagiarize. People don't know they're doing it, but if you pick up a sentence from a literature, verbal term, put it in quotes and put a reference. Okay? Because some journals might come back and tell you that this is very similar to the other. Believe me, then check up to this paper, and then you can change it. Okay? So, I think I've gone through all this, so this is fine. Okay, this is, I don't know if you have confusion when to use which and that. Have you ever, when you're writing to a non-English speaking person? So this is, use that for defining, like, tell us which one. We will select the option that measures glucose levels. When you're using which, it's for non-defining. It's a fact about a known option. So here you have, we will select option A because you know what this option is, which measures. See the difference? In the first one I didn't know. I used that. But when I know the option, it's which. Okay? And do not use double negatives. You know what that means?
Don't use no double.
I've read papers that says it was not, and not, something like double negative is saying that not, it isn't happening twice in a different way. Don't use that. Okay? Okay. This is all. Okay, remember to never split the infinitive. You know what that means? So here is a sentence. She seems to really like it. You don't split this. Actually, it says she really seems to like it. See the difference? Okay, so you never split the infinitive. It's always next to the subject. Okay? And this is, they say, okay, I don't know whether you understand, but if you say and, or, in science you don't use that. Lawyers use it all the time. Okay? And never start a sentence with because. Okay? English language doesn't like a sentence you starting with because. Okay, so these are some English or style suggestions. And I think with that I can complete then. Okay, this is it. So the writing process, you start out broad and then you publish, you achieve the end, okay? Okay, I think that's it. Now you feel you can write a manuscript? Adriana, you're going to get manuscripts tomorrow from her.
I realized